Analysis. So for those who are interested, hello everyone, can you hear? Okay, I will start now. Can hear, right?
Yes, can hear.
Okay, a very good afternoon, everyone, and welcome to INBIOSIS webinar lecture series, the 13th lecture series for this year 2021. I am Dr. Emelda Rosseleena Rohani, will be the moderator for today's webinar. I hope the video has provided you with some background of UKM and INBIOSIS. So for those who are interested to collaborate with us, you are most welcome to do so. Please feel free to browse the INBIOSIS website and social media for the latest updates and information. I feel so grateful today that all of us are granted good health and time to attend our webinar series. There will be many more webinars to come, and I hope all of you will join us again in the future. INBIOSIS webinar series is established to provide a knowledge and research experience sharing platform to ensure we are still connected with each other and stay motivated in our research despite this saddening pandemic COVID-19. Without further ado, I would like to introduce our honorable speaker for today, Professor Dr. Sahidin, from the Faculty of Pharmacy, Universitas Halu Oleo, Indonesia. Allow me to share with you Prof. Sahidin's CV. Okay, Professor Dr. Sahidin is a professor of natural products chemistry. He obtained his PhD in the Institute of Technology Bandung in 2006. He did hold a postdoctoral position in the Faculty of Science and Technology, and also INBIOSIS UKM from 2009 to 2010. Currently, he is a lecturer at the Faculty of Pharmacy, Universitas Halu Oleo, Kendari, Sulawesi Tenggara Province in Indonesia. His expertise is in isolation and structure elucidation of natural product compounds. He has a vast research experience in isolation and structure elucidation of natural products compounds from Pterocarpaceae, specifically the genus of Hopia, Shoria, and Vatica, Euphorbiaceae from the genus of Jatropha, and also Polygonaceae, specifically in the genus Polygonum, or in specific the Cursum species. He also worked on the Zingiberaceae, on the genus of Alpinia, Etlingera, and Mysteria, as well as several other sponge and soft coral in Indonesia. So without further ado, let us welcome Prof. Dr. Sahidin to share his research findings. The floor is yours.
Okay, thank you very much. Assalamualaikum warahmatullahi wabarakatuh. Thank you, Dr. Emelda, as a moderator. I permit to share my PowerPoint, okay?
Yes, please. slideshow. Okay.
Yeah, can you see? Okay, ladies and gentlemen, thank you very much. Especially the first, I would like to thanks to the INBIOSIS officer. The first, Prof. J.T. Ajura as the director of INBIOSIS. Professor Ain Sarunata Kain is deputy director of INBIOSIS. Colleague in INBIOSIS, maybe next time I can visit INBIOSIS again. Special thanks for former director INBIOSIS, Prof. Norma. He has provided me support and become a role model in development of my research. I do hope that she always be healthy and happy with her family, Prof. Norma. In this webinar, I just want to share my research activities at Faculty of Pharmacy, Universitas Halu Oleo. I hope there is something that can be followed up together in the future. The paper with title Chemical and Pharmaceutical Aspect of Etlingera, Etlingera Plants, Isolation and Structure Elucidation. I am Sahidin from Faculty of Pharmacy, Universitas Halu Oleo, Kendari, Indonesia. Before I continue my speak, I want to introduce the location of my hometown in the territory of Indonesia. This is Kendari here. Kendari. Kendari is a capital city of Southeast Sulawesi province. There are two routes from KL, Kuala Lumpur to Kendari here. You can fly Kuala Lumpur, Jakarta. In Jakarta can directly to Kendari, or you transit in Makassar, and Makassar to Kendari. This is the first route. The second route, you can use for flight for Kuala Lumpur to Makassar and Makassar to Kendari. I hope next time you can visit my hometown. This is a good view in sea for both snorkeling and diving. This is map of Sulawesi Island. Okay. Sulawesi Tenggara province has 651 islands. This archipelago province, 391 is named and 290 unnamed. 75% is the sea. So the consequence for that, they are abundant natural resources, both marine and land of the province. From the land, based on the books of Etlingera of Sulawesi, our research focus on chemical and pharmaceutical aspects of Etlingera, especially Etlingera growing in Sulawesi. You can see that the distribution of Etlingera in Sulawesi, this black spot, this is a location, the Etlingera. Why our group study on Etlingera? The first, 48 species of Etlingera grow in Sulawesi, and 14 species is growing in Southeast Sulawesi in here. There are 14 species. And the Etlingera easy to get because growing in the secondary forest. For the publication, until now, the very limited publication, both chemical and pharmaceutical aspects. This is the opportunity to get a new compound, maybe Q1 publication, and so on. And the people use as a traditional medicine plant, so safe for to consume, and Etlingera produce volatile and non-volatile compounds. In the list, this is a species of Etlingera growing in Sulawesi. Until now, from Sulawesi, only 2 species have been studied. Etlingera elatior, I think this is popular in Malaysia. Bunga Kantan, I think, the name in Malay. And Etlingera is endemic of Southeast Sulawesi. To get know the Etlingera more closely, I will explain biological and compound from the Etlingera. This is two species of Etlingera from Sulawesi, Etlingera elatior and Etlingera arophyll. Means the bunga is flower and buah is fruit, batang lepisode, rimpang means the rhizome, and daun the daun means the leaves. This is a differences between Etlingera caropis and Etlingera elatior. Until now, I have reviewed about less than 20 species of Etlingera have been reported, and the most interesting for the compound is produce a derivative of curcumin. Curcumin, I think the popular compound, and in Gajah Mada University Yogyakarta, there is a research center special for curcumin research center. And from Etlingera, we can find the demethoxycurcumin, heptathione, and this compound. This is the group of the aryl heptanoid because the compound has 2 unit aryl like this, and 7 compound as a bridge, 7 carbon as the bridge. So the compound is group of diaryl heptanoid because content of 2 aryl groups and bridge atom carbon, 7 atom carbon as a bridge. The second group I think is this is the phenylpropanoid. This is a common compound like paracumaric acid, capionyl coenate acid. This is a common compound. And the flavonoid also a common compound like catechin, isoquercetin, quercitrin, quercetin, caemperol, and so on. I think this is the common compound, common flavonoid. And some of another compound is steroid group, stigmasterol, beta-sitosterol, stigmas-1, and there are 6 steroid from Etlingera. The Etlingera also produce phenolic and amine, but still limited, and 2 diterpen labdan group. Like other Zingiberaceae, Etlingera produce a volatile compound. There are many volatile compound. Part of them like this, like decanal, asam decanoic acid, hexane, and so on. This is a volatile compound produced by Etlingera plants. Based on biological activities of isolated compound of Etlingera plant, if you see this table is the biological activity is still limited. No more reported of biological activities from Etlingera plants. So I think this is the chance for me, for us, for someone who study of Etlingera to do collaboration on biological activities essay. One of the problem why the biological activity is still limited, one of the problem is that the number or weight of compound that have been isolated is very limited. So that is not enough to carry out various biological activities of for the test. This is a challenge for me, for us, for our group, to isolate compound in a amount in the bigger from other biological activity. So in this case study, I want to show isolation and structure elucidation of secondary metabolites from Sulawesi Etlingera using semi-bioassay guided, not bioassay guided completely, but semi-bioassay guided. In isolation, the most important step is to eliminate non-target metabolite, because we usually use silica gel as a adsorbent, and its polar is a normal phase, so it's difficult to separation a peripolar compound. So we have a like pattern. If we work with leaves, first separate the chlorophyll. So the chlorophyll will stick in the bottle. If the extract ethanol or methanol, add the water. Chlorophyll will stick in the wall of the separation panel. And if we work with the bag or steam bag, first separate the tannin and lignin using the ethyl ether like this. This is from the ether fraction, and this is methanol or ethanol residue. And for fluid or the plant contain sugar, the first separated the glucose acid combined using ethyl acetate. This is a glucoside, this is a fraction of ethyl acetate. And if you use with marine resources, the first we must free from salt using acetone and ethyl acetate. This is my pattern in our laboratory to get more metabolite secondary metabolite. This is I think this is I have a personal experience about this method. When while I work with Kesum in INBIOSIS, at the first 6 months, when I fractionated using vacuum liquid column chromatography, the sample I prepared about 20 grams. But the total amount after calculate in here, I just 2 or 3 grams. So my question, where is the 17 grams? This is make me frustration. My prediction, the 17 gram in adsorbed by this silica. This is my hypothesis, and I take, so to solve this problem, I take the test tube, I add the sample, and add the solvent. All of those solvent in the lab, I try to add to the tube. As a result, the addition of ethyl acetate formed 2 layers like this. I repeat this little discovery, then I analysis using TLC like this. This is a lower part, and this is the upper part, using ethyl acetate, and this is the total. After then, I analytically finally believe that 17 grams is stuck in the column with the small impact. Instantly, it's possible to save energy, to save silica, and to save solvent. This is a little finding when I work in INBIOSIS. Thank you, in INBIOSIS. And this method I use until now in my laboratory. So after get this pattern, we can publish in scientific report and finding the polygenomes. Okay. This method is developed by my student, Elias. This is a general procedure of isolation secondary metabolite from Etlingera using bioassay screening, semi-bioassay screening. The first sample extraction usually using methanol, ethanol. We got extra, and fractionation using vacuum liquid chromatography. We have 5, 6, or 7 fraction. And here, bioassay guided, here we try using a bioassay, and the most active fraction we continue to get a pure compound. A part of pure compound we use for structure elucidation using FTIR, NMR, MS, optical rotation, and another using for bioassay. So we will get compound and molecular activity. And the challenge for other scientists or science expert, I think, how to increase concentration of the active compound in the sample in the plant using tissue culture and so on. This is a case study isolation of active compound from Etlingera. The first extract ethanol about 270 grams. Add the ethyl acetate and fraction ethyl acetate about half of the ethanol extract. This is a profile of ethyl acetate. We use vacuum liquid chromatography like this. Is simple facility, simple tools. We get in here 6 fraction, and all of the fraction analysis by TLC like this. This is the spot of the compound, and our task to separate this compound. Okay, this is using ethyl acetate hexane 91, 55, and 19 like this. After that, we test the biological activity, and the most active we continue for isolation. And here the most active is fraction B with weight about 4 gram. This B. This is for next isolation, and the others is we will do another time. From one fraction about 4 gram with a profile like this, we have success to get 10 pure compound. With wax like this, and compound 4 is the wax for 4. This is the profile of the 10 compound like this, all of the pure compound. Why is like easy? Because the first time we eliminate the untarget compound. So the next step is like easier. The next structure elucidation, we use spectroscopy because the instrument is I must join with another laboratory in Indonesia. So for known compound, I just check the NMR1D, both proton, carbon using depth analysis, and compare with the same data from reference. For novel on unknown compound, add the NMR2D, HMQC, HMBC, NOC, MS, and optical rotation. This is for a new compound or novel compound. This is isolate 4. From the proton NMR, we can conclude that the isolate 4 has 24 proton, 23 proton attached carbon atom, and 1 proton as hydroxyl. And based on carbon NMR, we get conclusion that isolate 4 has a 20 atom carbon. The atom carbon consists of 6 methylene, 1 methyl, 8 methane, and 5 quaternary atom carbon. So the total 20 carbon atoms. And this order, compared with the reference, isolate pore is identical with yakuchinon A. So we can get conclusion, isolate pore is a yakuchinon A. The same method in determining the structure of isolate pore, we apply it to another isolate, and we can elucidate 10 compounds like this: compound 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. And compound 1, 3, 4, 5, 7, 8, 9 is derivative of curcumin. This is interesting to do structure activity relationship, and this is one data of IC50 using MTTSI to cell lines. So you can see that the aryl heptanoid has a activity better than another. They have a activity better another compound, another group compound. Now, what is the big challenge for me, for us, for our group? How to do isolation is efficiently. We inspired by this price list of natural product compounds, where from Catharanthus roseus in Indonesia, the name is tapak dara. The scientists found a new two compounds, pinclistine and pinblastine. The differences in pinclistine contained aldehyde groups, and pinblastine contain methyl. But the price list, you can see that pinclistine about $2 million per kilograms, and for pinblastine about $1 million per kilogram. This is interesting for us to make maybe company for a natural product compound. This is new challenge for us. For our colleague is interested in isolation and structure determination. We try to rationalize the high price of pinclistine. And here this is a chemist. You can see that in pinclistine there are 9 carbon chiral, 2, 3, 4, 5, 6, 7, 8, 9 carbon chiral. So the probability to get this compound only 0.2%. If the compound get by regular synthesize. So how about the opportunity through isolation from matter directly? If the pinclistine concentration in dry leaves Catharanthus roseus only 1 ppm, means 1 milligram per kilogram dry leaves. So 1 kilogram dry leaves need 20 plants of Catharanthus roseus. So 1 kilogram pinclistine need 1 million kilograms dry leaf is the same with 20 million plants of Catharanthus roseus. Is very, very impossible maybe. And plant cultivation, if one plant need 1 meter square, so 1 kilogram pinclistine need 20 million meter square or 2,000 hectares. So this is challenge for tissue culture expert, how to increase the content of pinclistine in the leaves of Catharanthus roseus. Maybe the first 1 ppm, the next time to be 5 ppm, the next time maybe 100 ppm, and so on. This is a new challenge in science. And from INBIOSIS, when I work in INBIOSIS, we have isolated new compound, and the name is polygonominous, A for polygonominous. The compound has cytotoxic against cell Q562, MCF7, HCT116 cell lines, and as a HIP1 protease inhibitor. This is a interesting potential of polygonominous A. So how to increase a concentration polygonominous A in the lips of a polygonominous? As a conclusion, the Etlingera plants produce a variety of compounds, including the ideal heptanoid derivatives, polypropanoid, flavonoid, and steroid. However, the study of its biological activity is still very limited. The availability of pure compound with the right structure and large quantity is needed for testing various biological activities and other purposes. The very high price of active compound is a challenge and an opportunity to provide raw material with biotechnological enrichment of active compounds and to isolate this active compound. Thank you. 10 years ago. Okay. Dr. Amel, time is yours. Assalamualaikum warahmatullahi wabarakatuh.
Thank you very much, Professor Sahidin. What vast isolation work that he showed just now. I still remember our days in the lab that we have to stay back until night to isolate the compounds and from kilograms of samples, right? So there is a laborious work to do the isolation part. Okay. Let's see that if we have any questions, but I think I will start to make the ball rounds first. Okay. Well, would you like to explain more on how do you do the semi-bioassay guidance? Before it's only the based on bioassay guidance only, right? So what is the difference? What is this semi-bioassay guidance?
Okay, thank you very much. I am a natural product chemist. Usually, I didn't use a bioassay. I just isolate the target is new compound. All of the compound I must isolate of them. So active is inactive is just a complement for me. So because I work on my affiliation in pharmacy, I must combine the bioassay guided and phytochemist guided. So in the middle, I just need a reason why I choose this fraction. So I try to using bioassay. So maybe the name is yeah. I just give the name. This is a semi bioassay guided like that.
Ah, okay. So you showed the plant just now, tapak darah, right?
Yep.
Yeah, Catharontis roseus. So in Malaysia, I think it's called kemuning Cina. So the compound vincristine, it costs 2 million USD, right? USD per kilogram. So until now, they still have to extract the compound or already synthesized. There's already synthesized compound for vincristine.
Until now, some people say this is a semi-synthesized. So apart raw material from isolation from the natural, apart maybe I will show the like this. I get a paper like this. In here, depending to the part, the one part is from natural, and another from the natural. Just the changes that combine this here in with here, semi-synthesized. Because it's very difficult because the pyridine has 9 atom carbon chiral. So the stereoisomer about the compound like this, has the same physical and chemical properties about 512 compounds. So the Princeton only one compound from 512. One of 512 is a Princeton. If to get the compound using a synthesize, fully synthesized.
Okay. Another question from me. Is it okay? uh, you shared just now the say A
Polygonal.
is extracted from the stem, right?
Yep.
Of the kesum plant. So this means that polygo polygonu means A,
Yes.
right? So it doesn't. We didn't find it in the leaf, is it? So this compound is only from the stem, very highly.
Okay. I think if the compound has a complete structure, you can use this compound as a standard. You can analyze the leaves, the rhizome, the stem using polygoniums in standard. So from all of the part, you can see a concentration of the polygoniums. This is can
Yeah,
using as
because
a standard.
yes, yes, okay. Because we consume mostly the leaves, but so now we have to include the stem as well. Okay, let me read the questions that we have here. So welcome, Dr. Nur Khalida. This is question for you, Prof. Assalamualaikum. Thank you very much for your very interesting talk. May I know if you have any tips or method that you use for crystallization of pure compound? Any tips atau method?
Recrystallization usually compound group triterpene or terpenoid can get in crystal forms, and usually thing is just like fortune yeah, to get a crystal in isolation. Usually terpene compound, but another compound, especially phenolic compound, sometimes get in a crystal, usually in an amorph form. So I don't have a strategy on research how to get make a crystal from the natural product compound. I'm sorry.
So you normally isolate the pure compound but doesn't go further to crystallize, is
No,
it?
no. Just get the
Okay.
pure compound.
All right. And then send to NMR.
Yep. And so difficult to send to X-ray crystallization.
Yes. Okay. All right. So another question from Dr. Muhammad Shamsul Reza. Assalamualaikum. Hey. Okay, Prof, do you need a different technique when extracting compounds from plant or sponge from the sea? Soft coral from the sponge, compared to extracting compounds from plants on the land. Is it there's a different? Is it any different technique to extract compounds from sea and then compounds from the land?
Yeah. Theoretically, salt from salt is unsolved in the methanol or ethanol. So salt just solved in waters. in theoretically, but reality in the sea there are many organic salt. So usually if I use methanol, ethanol, it's very difficult to continue because there are many salt. After I work about 2 or 3 years, work with a marine organism, so the most efficient, the first extractant you use, you should use acetone or ethyl acetate. Is better than using ethanol and methanol. And the other step like fractionation using vacuum liquid chromatography and radial chromatography, I think is the same. But in the preparation is different.
Okay, all right. Okay, so question from Dr. Wan Muhammad Aizat. Thank you, Prof, for the interesting talk. Does Prof consider to use synthetic biology technique to produce the vincristine?
Pardon me?
Do you consider to use synthetic biology, biology synthetic, to technique synthetic biology to produce the vincristine?
Yeah, I hope there are many experts in synthetic biology to overcome the problem, how to increase the vincristine from the cataratus residues, maybe at the Elucidation and so on. I don't know, I just want to give a issue like that. Idea like that.
Uh-huh.
I don't know how to about proteomic, transcriptomic. I don't know. Just I have idea like that.
Okay, thank
idea.
you. Okay. Okay, next question from Dr. Sharuna Takain. Do you think is that possible to chemically synthesize polygonumins A? Chemically synthesize.
Yep. I think polygonumins A, this is consists of sucrose, sugar, like this, and phenyl propanoid. This is theoretically this is just esterification. Phenyl propanoid with sucrose, like that. But I think this is there are some active position like this OS. This is can make esterification like this. So I think this is must use protecting groups for to protect this in. Just this is, I think this is possible. Okay, it's not too difficult. I think to synthesize this compound.
I think this.
Because
So
the
this is a
basic is
good news.
sucrose. This is the basic is sucrose and phenyl propanoid.
Okay,
Maybe I hope this is. I am optimistic. This is can success with synthesize regular synthesize in laboratory.
Although the structure is quite large. So I think this is good news for Dr. Ain. So for further study, right? Okay, so another question from Dr. Nur Khalida. What is your strategy for structure elucidation if you have only, if you have pure compound of 1 milligram only?
Yeah, 1 milligram. Sometimes 1 milligram. If the like instrument like NMR, it's okay. All of the instrument is okay. I think it's the same strategy. Okay, but the problem 1 milligram like noise in the NMR. Very difficult to analyze only 1 milligram. So the strategy how to increase the number of your compound, not 1 milligram. Maybe 5 milligram is okay.
So we must increase the pure compound,
Yeah.
right? To at least 5 milligram.
Yeah. So my strategy how to isolate in the big number of compound.
So what is the, can you share with us what is the strategy? So start with the large number of raw material, is it?
Yeah.
And then choice of solvent.
Yep.
Okay.
Choice.
All right. Any questions? Wait, let me check. So any question, anyone? All right. Basically, how long it takes to purify the compound?
In in
In
UKM, I
general.
think it's okay, yeah, because the solvent in UKM, especially solvent for make eluent, is using scientific grade. But in our laboratory using industrial grade. So in UKM, I think it's better work than in my laboratory in Indonesia because I must purify the solvent. The first step to make purification of the solvent.
I see.
I make a distillation.
Oh, okay. Extra step to do
Yep.
in Oleo.
Yeah. If one on 2 liter is okay, but one 100 liters is very high too to make pure the solvent.
Okay, so please come again here to continue your work, to continue doing work here happily.
Insya Allah.
Inshallah. Okay. All right. I think let's see if we wait for several minutes and see if there is another more questions coming in. So any advice? Yeah. Any advice for those who are doing natural product, Prof. Sahidin?
Yeah, maybe not only in Indonesia, but in Malaysia too. I think maybe conventional isolation. Some scientists is not
Not
scientists
prefer.
usually like using a high technology instrument like metabolomic, but for isolation, some scientists avoid this process. So I think this is a challenge for me, for us, maybe to back to the conventional isolation. I think you will have an experience like that. Work from 8 a.m. until 12 p.m. every day.
That is why people don't want to to venture into this hard work. Yeah. But do you think this conventional technique in natural product is still relevant for nowadays research?
Yes, because some new compound can stop without isolation.
Okay, we have another question. From Ririn, other than bioassay guided isolation, what methods can we use for isolation?
Usually see bioassay guided, for example, my question when I do co-promoter in Pajajaran University, the promoter say usually the finding of active compound as antioxidant, for example, only get quercetin and quercetin. So quercetin is active compound for antioxidant. The most active in sample. This is maybe I am not too interested bioassay guided because the compound just like that. But for chemist, for me, I look for new compound. All of the compound in a plant, I must isolate them. 9, 10, more 50 compound, I must isolate to get new compound. Like that. So I'm not interested, not too interested work with bioassay guided.
In the bioassay. That that is why I think it's important for networking so that the other group will do the bioassay site, right? Because your group will only do the or concentrate on purifying new compounds. Am I right? Okay. So there's a next question. After being an extract, what is the best separation method? Liquid extraction or directly with chromatography such as column and others?
Yeah. Usually with liquid extraction, this is for in my laboratory is for pharmacology is like that. But for me is usually directly using fractionation using column because maybe liquid chromatography, one sample to be two fat, upper and lower.
Repeat again, one sample.
One sample can divide into two fat.
Oh, yes.
Two fat, one and
Yeah.
two. This is
Two
extraction
phase.
liquid. Yeah. But using a chromatographic like column chromatographic, we can the sample divided into 5, 6, 7 fat. So it's more simple.
So which she was asking which one is the best separation, the
Yeah,
liquid
using
or
a chromatography
chromatography?
directly, more
I
efficient,
see.
more efficient and faster.
Because can directly get 5 or 6 groups
Yep.
from the chromatography. Okay. For liquid, we can only get 2, the upper and lower, right? Okay. Okay. I hope this answers the question from Rivin. Okay, another question here. May I know how to remove the lipids and tannins in the extract before further to column chromatography? Because the lipid can clog, sumbite can clog the column sometimes.
Lipid usually as a non-polar compound. Using n-hexane, I think it's okay. And tannin, this is a very polar compound. I usually use diethyl ether.
The
Like
diethyl?
Diethyl ether.
Diethyl ether. Diethyl ether.
I think in the
Okay, so
basis I have bought diethyl ether in the aluminum foil, the bottle like aluminum foil.
Yeah. Okay, so I hope this answers the questions. I have another question. In general, can you recommend how to isolate alkaloids?
Oh, alkaloid. Yep. Yeah. In the natural alkaloid, there are two positions. First, as salt, alkaloid salt, and the second is free alkaloid. For free alkaloid, I think can isolate directly, but from salt alkaloid you must free to make the free alkaloid using like acid and so on. To
Acid base extraction. Oh, all right. Okay, I think are we are finished with all the questions. Oh, another question. Pak Sahidin, you have a lot of questions. Thank you for your answer, Prof. Previously, I separated with liquid extraction. This is from Ririn. She asked before the one on the liquid extraction. But why the extract top layer and bottom layer often have the same profile?
Yep. Yes. Like
Have the
that.
same TLC profile.
Yeah. Sometimes like that. This is because extraction liquid not specific. So sometimes like that. After analysis using TLC, the profile is the same. This is
Oh.
this is a problem because maybe some compound solved in the ethyl acetate and another part solved in hexane, two layer, but with the same, just different only concentration.
So what is your suggestion?
Yeah, my suggestion is continue one of the fraction, usually the more polar, semi-polar like ethyl acetate and hexane can continue by chromatography. Use a vacuum liquid chromatography is better and faster.
So Professor Sahidin suggested to use the chromatography for more efficient isolation of compounds. Okay. So thank you very much, Prof. Sahidin, for your sharing on your experience in the isolation, especially, and also the structural elucidation of compounds, of structure compounds. Okay, so let's move to the next session. I hope everyone satisfied with the answer. Okay. So I would like to thank Prof. Dr. Sahidin for the very interesting subject that I am sure has opened up our view and give you an idea on how you want to bring up your research output to the next level. So today's webinar had recorded around, wait for a minute. Around 48 participants in WebEx and 14 participants in FB Live. I would like to thank all participants for spending your time today. Please stay safe and take very good care of yourself. Before we say goodbye, let us immortalize this session by switching on your video and have our face captured. Okay, so take care everyone and see you again in our next webinar. Professor Sahidin, please wait, don't go anywhere yet. Okay, thank you very much.
Thank you very much. Ibrahim, I think, the attend of Ibrahim Jantan.
Yes.
How are you, Prof?
Okay. Everyone can on your video so that we can take a picture together.
Prof, when will you come Kendari again? Prof Ain. Sehat-sehat.
Okay, I will pass to Intan.
Okay, everyone ready? Smile. 1, 2, 3. Okay, wait a minute. We'll take another one picture. Okay. Okay, ready? 1, 2, 3. Okay. Thank you, everyone.
Okay, thank you. Stay safe. Bye. Please wait for a while for the attendance at the end of webinar, at the end of this video later on. Assalamualaikum Pak, boleh masuk breakout session.
Amel?
Tadi tak boleh nak invite.
Oh,
Sekejap saya masukkan semula.
masih ada ini.
Sudah masuk?
Saya ada program ini. Saya boleh communication dengan ibu.
Boleh dengan Dr. Ain?
Boleh. Belum? Mana ada?
Belum. Okey, sekejap. Mana ni? Kenapa? Sebentar ya, Pak.
Kan saya
Sudah
sholat
maghrib.
asar. Asar saya
Oh,
sudah.
okey.
Nah, lah. Karena belum asar ya?
Belum.
5 petang ya?
4 lebih lah. Ada orang mahu bercakap dengan bapa, jadi sebentar ya.
Ini di rumah, Mel.
Di office.
Oh, di office. Kita boleh masuk office.